OpenProt is the first database that allows a polycistronic annotation of eukaryotic genomes, recognizes the coding potential of pseudogenes, and enables the discovery of previously undetectable novel proteins.
We have designed this protocol so that it is accessible to all users without requiring extensive bioinformatics skills. It essentially places proteomic discoveries within anyone's grasp. To comprehend and tackle today's challenges in medicine, we need to fully understand the proteomic landscape and all of the actors and their dynamics. OpenProt provides that possibility.
Although here OpenProt is used for the analysis of proteomics experiments, it can also be used with other systems, as it simply provides a more accurate definition of the proteome.
Begin by opening the OpenProt website and using the link from the top page menu to open the Downloads page. Click on the species of interest based on the analyzed experimental data and the desired protein type. Click AltProts, Isoforms, and RefProts to generate files containing all of the known and novel protein types present in the OpenProt database, and if available, click on the annotation from which the protein sequences are drawn. Click on the level of supporting evidence necessary for the protein consideration according to the research objective. Then click All Predicted to generate files containing all of the OpenProt predictions, and click the desired file format to download. For proteomic analyses, select the FASTA protein file. The README file will contain all of the necessary information on the file format. For database handling, log into